So you've chosen to study A-level biology. Great choice. You are going to love it. It's a big step and be prepared to work hard and be challenged, but you will be rewarded with even greater insight into how our amazing natural world and bodies work. In this video, we're going to give you a head start to ease the transition to A-level by consolidating core GCSE knowledge needed for Year 12. We're also going to throw in a few nuggets of A-level knowledge in advance, which you will dive deeper on in September. We'll work through concepts in the order you should expect to see for AQA. However, all of this recap and knowledge will stand you in great stead for EdXL, OCR, or WEGEC also. So let's start with cementing our knowledge of biological molecules. But before we get going, I want to draw your attention to the free download in the description on common misconceptions in biological molecules. I made these for all chapters for AQA A-level for my students to summarise common misconceptions, terminology errors, and examiner comments on the topics. Every year, when the examiner reports for the summer exams are available, I update these resources. The idea being my students have the examiner feedback in a much more digestible format. If you've ever tried to read an examiner report, they are quite hard going to read, and the valuable information you find in them is often lost amongst a lot of examiner blurb about how well questions differentiated students of different levels. So I would have a look, download it, and use it as you work through the biological molecules chapter. And if you find it useful, you could choose to download some of the others available individually or as bundles. They get updated every year, but once downloaded, you will always get free access to any updated versions available. Now, first up in biological molecules is carbohydrates. You should remember that carbohydrates are our primary energy source in our diet, and that is because many carbohydrates are polymers, long chains of sugar monomers, and enzymes called carbohydrases are able to digest, which means break down into smaller molecules, so we have glucose to use for aerobic or anaerobic respiration. Both use glucose, remember? So let's have a closer look at glucose. And as you should recall, the chemical formula is C6H12O6. You will learn to draw glucose in its displayed formula, as you can see here. Now remember, this shows the bonds between atoms, so we can distinguish single and double bonds, and it shows the arrangements of the atoms and functional groups. Now, in these more complex molecules, you will notice we don't always show the carbon atoms. Molecules that contain carbon are called organic molecules, and all of the important molecules you'll learn about, DNA, RNA, proteins, lipids, ATP, all contain carbon. So often we just show it as a join, and each carbon is given a number representing its position in the molecule. And when these monomers bond to make polymers, these numbers are really useful shorthand for explaining where the bonding is occurring between monomers. You will learn lots about different sugars and carbohydrate polymers, some of which are made of this version of glucose, which we call alpha, and some are made of an isomer of this called beta glucose. If you remember your physics term isotopes, these were atoms of the same element that had differences in mass due to the number of neutrons present. Isomers are chemical substances that have very similar but slightly different arrangement of atoms in, and you will see in beta glucose our hydrogen and hydroxyl group on carbon 1 are flipped. When you learn more about carbohydrates in September, it's really important you ensure you can describe the structure or property a molecule has and be able to directly link it to how it is useful for its function. Just like when you've described adaptations of tissues or cells, you've had to describe a feature and link it to how it helps the tissue or cell perform its function. The sooner you master the importance of making these direct links, the quicker you will pick up more marks in exam questions. So let's move on to lipids. You'll hopefully remember from GCSE that lipids can be broken down by the enzyme lipase into fatty acids and glycerol. And again, you're going to need to identify these molecules from their displayed formula and potentially draw a displayed formula. So if we take a closer look at their chemical nature, you will see we have a carboxyl group on the fatty acid chains, and the rest is made up of hydrocarbon chains, sometimes with double bonds between carbons and sometimes single bonds. From GCSE, you should remember that we need lipids for energy and insulation, but actually lipids are incredibly important in building cell membranes also. You will learn about these two different types of lipid, triglycerides, which are made of glycerol and three fatty acids, which is a great molecule for energy, and phospholipids, which are made of glycerol bonded to two fatty acids and a phosphate group. And the chemical properties of these molecules are crucial for forming cell membranes. So next up, we have proteins. And this is a hugely important topic for A level because you will remember enzymes are made of proteins. And enzymes are involved in so many fundamental processes you will learn about across A level. From GCSE, you will probably be able to name lipase, amylase, a form of carbohydrates, and pepsin, a form of protease. However, here are just a few more of the amazing enzymes you will become acquainted with as you work through the course. You should remember that proteins are polymers of amino acid monomers, and I have another displayed formula for you that you will need to learn to draw. This is the general structure of an amino acid. We call it the general structure because there are 20 different amino acids, all of which share the carboxyl group, an amino group, and a hydrogen off the central carbon. They differ, however, in their R group. R group refers to a variable group, and you will see this term used when referring to the hydrocarbon chains in fatty acids also, which can vary in length and saturation. Now, when you've answered questions about enzymes in your summer exams, I am sure you have written about the specific shape the enzymes' active sites have, which makes them complementary to their substrate. At A level, you will learn all about how all proteins, even structural ones, have a specific shape related to their function, and that this comes from different aspects of their structure at different levels in the molecule. Proteins are so important to organisms because they are needed for growth and repair, as well as enabling mammals to effect an immune response to a pathogen. They make up important enzymes involved in life processes, and they act as receptors for chemicals such as hormones or neurotransmitters to bind to allow cells and organs to respond to each other. So we're going to focus back on enzymes. Let's recap our GCSE keywords for enzymes, and I'm going to ask you to pause the video and write down or recall mentally some key terms or phrases you use when describing enzyme rate of reaction. The reason why I'm asking you to do this is detailed terminology is so key to gaining marks in all exam questions at A level. And you have most of these keywords in your vocabulary for enzymes already. So get them to the forefront of your mind and you are going to fly at this topic. Have a go. I wonder if you can get all 16 of mine. So, how many did you get? Hopefully, most of them. I need you to make a mental note of how important these terms are. Your first required practical will be investigating rate of enzyme controlled reaction. And you will remember our amylase practical from GCSE, where we tested for the presence of starch using iodine. And when the sample stopped going blue-black, we knew all of the starch had been broken down. The faster this happened, the closer to the optimum conditions for the enzyme. Now, you'll likely perform several more advanced investigations into enzymes at A level, using A level techniques and instrumentation such as serial dilutions and colorimeters. What stays the same is the graphs of enzyme rate of reaction and explaining how different factors affect them. So let's do some recap. Either side of the optimum pH, the enzyme's active site starts to denature. So it's no longer complementary to the substrate, meaning there are fewer successful collisions per second between the enzyme and the substrate. And this means fewer enzyme substrate complexes formed per second. And when we look at how enzymes are affected by temperature, at low temperatures, particles have low kinetic energy. So there are fewer successful collisions with sufficient activation energy, so fewer enzyme substrate complexes form per second. At the optimum temperature, the frequency of successful collisions and enzyme substrate complexes formed is highest. At higher than the optimum temperature, the enzyme's active site starts to denature, so there is a lower frequency of enzyme substrate complexes formed. Now, your A level teaching on protein structure will really give you a higher understanding of why this happens. The other two graphs you should be familiar with from enzymes at GCSE are rate of reaction when enzyme concentration or substrate concentration is your independent variable. As enzyme concentration increases, rate of reaction increases as more frequent enzyme substrate complexes are formed. But after a certain point, there is so much enzyme and you have more active sites than substrate, so the reaction is over almost immediately. As substrate concentration increases, the rate of reaction will increase, as there's a higher frequency of successful collisions until all of the active sites are saturated and working at full capacity. Here, the rate of reaction will plateau at its maximum possible rate. Now, in A level, you will learn lots about how these graphs might change when enzyme inhibitors are used. And I cannot tell you how many applied questions you will get where the examiners will tell you about an enzyme you haven't heard of and how it's inhibited by a molecule you've never heard of. All you need to do is strip away the peculiar names and connect the processes, which are or are not happening anymore, and how that might produce the results seen. Now, one skill from GCSE chemistry that I find students are still struggling with at A level is calculating rate of reaction. So, if you can secure this now, this will really elevate your performance in maths questions on rate of reaction at A level. You should know that the gradient on a graph showing time against volume or mass of product or substrate reflects the rate of reaction. The steeper the gradient, the faster the rate of reaction at that point. You should also recall that we can work out the average rate of reaction between 2 points by taking the change in y divided by the change in x. So on this graph, for example, the average rate across the first 40 seconds is 0.46 grams divided by 40 seconds, which gives us a rate of 0.0115 grams per second. Now, please look at how I've presented grams per second. At A level, you will more commonly see the slash removed and what was after the slash represented as a negative power. Now, if we didn't have a graph of data and only had a time for a reaction to finish, our iodine to stop turning blue-black, for example, we can calculate rate of reaction as 1 over time. And we use arbitrary units as we haven't got actual data to give specific units. Now, tangents are the other part that I notice students struggling with, still at A level. If you are asked to find the rate of reaction at a specific time point, you have to draw a tangent to the curve reflecting the gradient at that point. You must then pick 2 points far apart on that tangent to minimize error and use them to calculate the change in y over change in x, which will give you the gradient and therefore the rate of reaction at that moment. You can see here a couple of worked examples for the initial rate of reaction at the start and the rate at exactly 20 seconds. Pause the video if you need to follow the working. Comparing initial rate of reaction when investigating optimum enzyme conditions is really important at A level, because after the reaction has started, the substrate concentration decreases, and you will remember in enzyme rate of reaction experiments, substrate concentration is one of the important control variables which must be kept the same. Now, before we move on to nucleotides, I want you to pause the video and recall the food tests for protein, lipid, reducing sugars and starch. Very briefly, what is the method and what are the colour changes to and from? So you'll be delighted to hear there isn't a lot of extra knowledge at A level when describing food tests, other than a new test for non-reducing sugars and knowing how to gain a quantitative measurement using the Benedict's test, using a colorimeter or drying and weighing the colour precipitate formed. So let's finish off our biological molecules with nucleotides and nucleic acids. So you'll have learned at GCSE that DNA, deoxyribose nucleic acid, is a polymer of nucleotides, which are each made up of a sugar, a phosphate, and a base, and that the backbones of the two DNA strands are joined by hydrogen bonding between complementary base pairs, adenine with thymine, and cytosine with guanine, although you will have only been given the first letters of these bases. You should also recall that DNA exists as a double helix that can unwind to replicate DNA in interphase of the cell cycle or to be transcribed into messenger RNA during transcription. where one triplet base sequence in the DNA will eventually code for one amino acid in the protein formed during translation at the ribosome. Another nucleotide that you will have heard of but probably not realize it's a nucleotide is ATP, which is produced in respiration and is an important molecule for transferring energy. If we look a bit closer, you will see familiar features to the nucleotide in DNA that you've learned about: a pentose sugar, a base, and this time 3 phosphates rather than one. You will learn so much about the production of this molecule and its vital role in providing energy for biological processes throughout the course. So well done, you have just fast tracked your start to the course, which will make biological molecules a little easier. I hope you found it helpful and are looking forward to diving deeper in September. Throughout the course, do remember my channel is here with lots of help and support to really help you succeed. Explore the channel, see what other resources are available, and do share with others on the course. You can always comment if you have any specific questions or requests, and I will always do my very best to try and help you. So join me again to review key concepts in cells, transport across membranes, the immune response, and gas exchange to refresh essential knowledge ready to maximize your A-level learning.